Okay. So in our previous lecture, we were focusing on this idea of aqueous speciation, but as translated into a in vivo context. And so I was giving you the example of vanadium-4 and focusing on compounds that later on we'll talk more in depth about why these particular compounds exhibit any sort of anti-diabetic property. But anyways, the main focus here was that an understanding, it was an exploration to understand how these compounds potentially behave in solutions that approximate the fluid environment of blood. So this is an approximation, so obviously it's missing a lot of components. But in this particular speciation study, a number of important small molecules and also proteins were included. So I mentioned last time that they were using transferrin albumin as a representation of the serum proteins. There are more, but these two are one of the more predominant ones, particularly albumin, which is present at about 600 micromolar. It is the most abundant protein in blood and actually is one of the most important proteins in terms of the pharmacodynamics and distribution of drugs in general. And so also they included these series of ligands because of their capacity to bind metal and particularly the capacity to bind vanadium-4. So here on the right, were the list of, and I can go back so we can see that, were the list of ligands that have been explored when complex with vanadium have demonstrated anti-diabetic properties. So the idea was to look at these complexation systems that have a proposed structure like this in a solid state, but try to understand what happens when you introduce them into a solution environment that approximates the fluid of blood and under these conditions. So it's still a simplification in that we're not looking at all the things that are in the environment, but we're trying to get some of the features that might contribute to the dissociation of the compounds. And so this was just the assessment of what was observed in this particular study. So this is something in vitro, right? So this is in solution, a combination of different techniques to identify what compounds are actually being formed. And so generally what was observed is that a large portion of these compounds consisted of the vanadium center, so vanadium-4, bound to one of these bioligands. So that's either the proteins or the small molecules. And there was some indication of mixed ligation. So you have what is referred to as heteroleptic, meaning that you have two different ligands. So a combination of the original ligand plus one of these small molecules in this case. But the main feature, the main point here is that most of these compounds do end up dissociating and distributing the vanadium to these bioligands. So that's an important feature to understand the likelihood of the vanadium possibly being entered into the cellular environment where we're able to exhibit its effect. So this is to be able to see this a little bit with more clarity. We will go back into this later in the course to just dive in a little bit about what is believed to be the properties of vanadium that contributes to a sort of anti-diabetes 2 effect. All right, but what I wanted to just highlight was where I was at the very end of that lecture is what is the significance of this? So we want to understand speciation to, first of all, if we're designing a compound so that it actually has to remain intact, we need to understand, first of all, its solubility, okay, so, and as that leads to bioavailability, also the stability of the oxidation state of metal, whether or not that's important, if it needs to be locked into a certain oxidation state, or if it needs to be redox active in what type of environment. And then also to think about is general transport, biodistribution, but also the metabolic fate of it. So how it's metabolized and ultimately how it's excreted from the body. The idea is that you don't want anything to be long standing in the body. And so are there mechanisms to be released? So that's one of the most important features as far as profiling the general aqueous speciation of this. And so I mentioned that this is typically a pH-dependent speciation because we want to understand how this behaves over a broad pH range. So in the acidic environment, if you're thinking about designing something that might have a potential use for oral ingestion, or if you're thinking about just, say, an administration that's in the gut or IV, not IV, but intravenously, think about, well, what solutions that they come in contact, will it be able to reach the site of target? So with that in mind, just sort of an overview. So when we're focusing on metal-based drugs, we have that added element of the complexation of the metal itself and how long that covalent bond needs to last. So I've mentioned that a few ligands serve the purpose of an ionophore. So that is basically the function of delivery, let's say from outside the cell into the cell. So that basically is a delivery function. But there are certain instances where ligands are much more important than that in order to facilitate a certain activity of the metal. So that gives us a perspective of what may be the active species and what is the likelihood of that active species being able to reach a particular target. And then as we're going further along this idea of the biological fate, what are the metabolites that form? What are the effects of those metabolites, if any? And then again, thinking about route of administration, how do you propose this may actually be functional? And so again, what I have mentioned to you, this idea of being cleared from the body. Okay, what are the potential routes by which it can be cleared? Typically, when we're talking about small compounds, they're going to be filtered through the kidney. Something that we need to think about. All right. So we're going to continue this discussion about this balancing of metals. Let's go to next picture. And so what I'm going to start here is just a series of discussion about mechanisms of trying to control metals and this idea of disease situations in which you have a loss of that control. And then we're also going to look, and especially as we go into our discussion next week, a few studies, and so I sent you a homework assignment with papers that we're going to discuss that leads into this discussion about controlling metals and factors that can contribute to that. Get that right. All right, so as we've discussed thus far in the course, there are essential metals that are critical in terms of the types of functions that they play. We have been more focused on iron, and that's why our guest speaker was really diving into iron from the perspective of a cell death pathway that is iron, at least related, if not necessarily triggered by iron itself. And so a number of essential metals display that function by being controlled in terms of the amounts that enter into the cellular environment and that actually leave the cells. And so you see their function in the form of either cofactors or coenzymes for enzymes themselves, or participating in a number of important processes like the synthesis or even the breakdown of nucleic acids. So think about iron as it's important for the replication of DNA, as an example. And so there's also a space for non-essential metals, as I discussed in the very first lecture in this course, where there are a number of non-essential metals that are important because of their therapeutic capacity. So there's a certain concentration range in which certain metals do have this property. So these are a few of the things that I want to talk about in the over the next two lectures, including this one. So one of the things is that to deliver the correct levels of essential metals, in order for them to be able to engage in important biomolecular mechanisms, there exists a network of metal chaperones, trafficking proteins as an example of transferase. We talked about storage reservoirs as an example, ferritin, that's for iron. We've mentioned metallothioneins for copper and zinc. So all these play a very pivotal role in terms of trying to maintain that optimal metal ion equilibrium. And when there's either a slight shortage or slight excess that we've talked about, what mechanisms are imposed to try to get back to that optimal level. But often when there is an internal homeostatic environment breakdown, the body responds in a way that's going to be aberrant. So it's going to be responding in a manner that's not necessarily normal. And that's where you start to enter into a disease state. And then you have certain situations where the body will intentionally disrupt the metal ion equilibrium in a defensive manner. And we will look at this more later in the course with this idea of how the body has taught itself to protect itself against pathogens, and in a way that has to do with regulating certain essential metals. And also, we will look at an example of non-essential metal ions that are employed to restore a balance to disruptive processes. Again, this is a therapeutic function, but how that can play into trying to balance other processes in the body. And so there are examples where they are important, but you have to be really careful about how you regulate the amounts that are dosed into the body. Right. So we're going to look at an example where we're focused on this idea of an internal homeostatic environment breakdown, and a disease state that's a consequence of this. So in our first example that we're looking at is Alzheimer's disease. So this is a disease that affects older populations. It's a form of dementia. It's a neurodegenerative disease. So you can see the typical age range of people that do get affected by this normally. And so one of the symptoms of this particular condition, so progressive loss of memory, senses, and bodily functions. Incontinence basically means a loss of being able to control urination, excretion. So in later stages, patients experience difficulty speaking, eating, they lose their capacity to respond to the environment. So I just want to give you some of these statistics, the more recent ones that I can find. And so about 24 million people worldwide are affected by this condition, and that's expected to triple by 2040. In terms of expected lifespan, once you are diagnosed with this condition, or at least once it actually is onset, it's about 8 to 12 years. So here's the numbers. Number 7, global leading cause of death. This is for 2019 statistics. I tried to see if there's anything more recent. There should be something more. There's always a delay, right? So there should be something more for 2020, but I'll look further. But in Puerto Rico, that number is actually number 4 in terms of the leading causes of death. So this is the statistics. You're going to look at this more closely later on for Puerto Rico that I found, and these are the global one where it's not listed there, but it falls under number 7. These are just the top 5. All right, so when we're talking about Alzheimer's disease, obviously we're going to talk about something that has to do with the brain. So I'm just giving a few images as to where we'll be focusing on. A lot of here is where the condition manifests, but it's something that's actually going to be widespread throughout the brain in terms of the changes physiologically in the structure of the actual brain. And so an area that is going to be important to focus on in this area here, we have the ventricles and how that gets that, and also the tissue surrounding it, how that gets warped due to this particular condition. All right, so here's a cartoon representation contrasting on the left side a brain and the neurons that compose this brain, so these are brain cells, and what constitutes a normal brain, and then on the right side, what constitutes an Alzheimer's brain. So what we can see is a few different features. One is this idea of the atrophy, so how loss of the composition, but also the mass of the brain, but also you see these formations. This is a cartoon representation. So what are these formations? We commonly refer to them as amyloid plaques, but there's also things that occur within neurons that is a consequence of this. Here's another representation of where this is manifested. So here we have the representation of this neuron, and then what's happening at the membrane. So you have a really important protein. This is the amyloid precursor protein, this upper line is here, that's present. So that's a transmembrane protein. And what's happening is that protein becomes proteolyzed. So it basically gets clipped, and the products of that, some of them can start to aggregate. So you start to see these formations that form, and that forms around the neurons. And also, there's another feature that occurs within the actual brain, where you have these formations of this aggregated protein. This protein is called tau. And so you get this super aggregation that leads to similar types of conditions as far as what's observed for the amyloid plaques. So here it doesn't seem to be, there it goes. Here's just another representation. So this is a magnetic resonance image of brain extracted subjects. So we're looking at 2 patients. One is referred to as a healthy controlled patient. The other one is the Alzheimer's disease patient. So again, the same types of features that I had mentioned already. You see the atrophy of the brain tissue, and you also see this region here, the ventricles, that now have become enlarged, right? So there's quite a bit of change that occurs within the composition of the brain itself. So as it turns out, there is no cure for Alzheimer's disease. And so what has existed for a long time, and I've updated this, but I'm going to focus on in terms of just general treatments that are more commonly administered. They have to do with more trying to maintain the mental function, as much as you can, keep it operating, and managing some of the behavioral symptoms while trying to slow down the progression of the actual disease. So there are several FDA-approved drugs. Some of these are what is administered at early or middle stages. For instance, it's cholinesterase inhibitor drugs that help to slow down the symptoms, such as memory loss. Again, it's more for the maintenance of the health of the mind. And then you have these later stage treatments that are more palliative. So that's basically to help just to, for the conditions of the actual drug. It doesn't actually treat it. It just helps to just have some quality of life now that you've really progressed further along in the disease. So more recently, to 2021, the FDA approved this drug here, so Aduhelm, which was approved under this system, the accelerated approval pathway. So this is a pathway that is typically used for situations where you have a condition and there's not really many treatments available. So there's something that if it's promising, it can speed along the process of it actually getting used clinically. So when you are approved through the accelerated approval pathway, you don't necessarily need to have processed data from phase 3 trials. As I mentioned to you at the very beginning of the course, phase 3 trials are the ones where you're doing a much more extensive study, much more people involved. So when you're approved to this pathway, you don't necessarily, you may have done these type of studies, but you don't necessarily wait for those results and to get that out and to use. So this particular treatment is a monoclonal antibody that targets the aggregated forms of those amyloid plaques, of the amyloid beta. And it's observed to be able to reduce its actual burden, to try to break up some of that aggregation. So it's the first treatment that was directed towards the underlying pathophysiology of the disease, at least what's believed to be part of that implementation of that disease. And so as it turns out, 2 phase 3 clinical trials showed that the drug, so this particular antibody, had the capacity of effectively reducing the amyloid plaques. But the problem was only one of the 2 studies demonstrated that there was an actual reduction of the clinical condition, right? So that's 2 different things, right? Reducing the plaques, but actually affecting the health of the person, of the people. So once both studies showed, yes, you can reduce the plaques, but only one of the 2 studies showed that it actually reached that end target, the end point of reducing clinical decline. So it's very controversial, this drug. It actually faltered in terms of its clinical use in the US, and the company Biogen didn't even bother to release it in the European Union. So now it comes to just last month, 2023. So the FDA approved this other drug. Let's see if I can pronounce this, Lekemba, okay, to treat AD patients, again using this accelerated approval pathway. So this is a humanized immunoglobulin gamma 1 or IgG1 monoclonal antibody for the purpose of targeting the amyloid plaques and breaking them up. So very similar to the other antibody that was developed. So results from phase 3 clinical studies have been reported, but they have not been fully assessed. And so again, this was seen as a landmark discovery, but again, it's already mired in controversy because some of these results from these clinical studies have shown patients dying in these trials. As far as I saw, it was at least 3 patients, but I don't know total number of patients. Some of this information has not been released. And there's also questions about really how useful will this be? It's quite an expensive drug, and whether it can extend the brain health for a significant time versus cost and versus what the people have to be subjected to is not very clear. So the fate of this drug, uncertain. The other drug seems to be a bit of a dud. So that's where we are as far as the what's available for AD, but the title of this slide still holds true. There are no cures for Alzheimer's disease. So any of you are thinking about this, go ahead.
When it comes to them saying that there's patient death due to the drug, how do you decide that this is due to this drug and not just the fact that you have elderly patients with underlying?
Yeah, so I think that needs to be further assessed. But I think as soon as there's evidence of death, people start to raise flags. But yeah, that's something that needs to be fully assessed because those I don't think they've actually been published in a peer review. I think it's just that the data has been released. When you look at the clinical trials, you can actually find all the information about people enrolled and some of the maybe preliminary results, but not necessarily the assessment of it.
Okay.
Yeah. All right. So let's talk about what are some of the thoughts as to the let's say, the onset and progression of this disease. These are the things that are still a little bit hard to know, especially if with reference to the antibodies that have been developed to be able to treat the actual amyloid plaques, but not necessarily improve the condition itself. All right, so the biological cause of Alzheimer's disease is still something that is questioned, right? And there are several hypotheses. So there's one principal hypothesis, amyloid cascade. So what exactly is that? So that's the idea of these plaques that are building up. They are insoluble and just start to cloud the brain. And they're affecting that temporal lobes, that's the section of the brain that we referred to before, which is very important for memory function. So here's just this depiction of this idea of you have this protein, this amyloid precursor protein that's a transmembrane protein at the membrane, and you have these proteases that will clip this protein. And it's believed that these two peptides, so these are the peptides that are the small fragments of this protein, that are the ones that are contributing to this, this formation here. So you have two peptides that are basically virtually identical, A beta 40 and A beta 42. So when I say 40, 42, what I mean is how many amino acids are part of that. And we have it in sequentially here. So you see the labeling. So they're different by just two amino acids, the I and the A there. All right, so so it's believed that this section, which let me just clear this up a little bit, that this section here of these peptides contributes to this aggregation process because it can act, that's sort of the point of contact between the peptides as they start to come really close together. So it turns out individually, these peptides, the A beta 40 is quite soluble, whereas the A beta 42 is very insoluble. And so it's believed that because of that section, this self-aggregation can happen when there's a significant concentration of this loading up that leads to this cascade. That's why it's called the amyloid cascade hypothesis, where you have the monomers, so the individual peptides, then they start to aggregate at that point of contact, forming these oligomers. Then they start to form this extended structure, the protofibrils, and then what's more commonly known as these fibrils that form. And this is just a perspective of just structurally what's going on in terms of this, these peptides aggregating with one another. So here we just have to follow along the sequence of the peptides, just to get a perspective of how they basically form this type of structure. So the A beta 42 monomers will self-associate and deform into these two in-register parallel beta sheets. So you have this structure, you probably saw this in biochemistry, these beta sheet turn beta sheet motifs, and that's what is adopted by these particular peptides. All right, so as part of that cascade though, you also have a sort of a subset of that, this idea of metal ions playing a role here. So this is the metal ion hypothesis, but essentially that's building onto what we already has been proposed as an amyloid cascade hypothesis, where just the peptides themselves are contributing to this aggregation. The idea is that these metals, these essential metals, so iron 2, iron 3, copper 1, copper 2, zinc 2, they're contributing it by via coordination to sites in the peptides, leading to more extensive aggregation. Right, so that's what's going on here. There's an additional feature that's at least for the iron and the copper itself. All right. So again, hypothesis suggests that the metal binding to A beta influences the conformation of the two peptides and facilitates the incorporation into the fibrils that form the plaque. All right, so self-association of A beta peptides is easy and rapid. And so because of that, it can be quite difficult to assess the metal binding. Given that they're not necessarily the most homogeneous samples to work with, so you're trying to study metal binding with something that's not necessarily homogeneous, it can be very difficult. So people have tried to tackle this in a few different ways. Some early approaches is, well, why try to effectively lower the concentration of peptides? By doing so, the idea is that you're going to minimize aggregation, and so you have easier samples to work with to assess metal binding. But that then leads into additional problems where you can probably minimize aggregation, but then you're probably falling below limits of detection of techniques that can possibly gauge metal binding. So approaches that people have taken to overcome this to get some information. Well, let's get into that in a moment. But these are additional things that can affect this this makes it more complex to study. As we've talked about, pH being a factor in terms of speciation is very true here when it comes to the formation of these type of structures, and different factors like the ionic strength. So that's the amount of salts that are present. You have a really high ionic strength in your blend. It's about, you say, approximately 1 molar. If you want to give us a number. And so trying to understand how these factors and how to regulate that and how that actually makes it more complex is another reason why people have tried to simplify this study. And so what people have done, yeah, but this,
so when we're in this, I would think go around that and it's better than other means.
oh, okay. Yeah, I think the general solubility has to be within a certain concentration range. So probably an effective concentration, more of the peptides, you just really couldn't get it so that they can get some reasonable calculation. Versus the other one at a low enough concentration, you can see that it basically remains as a monomer. So probably some sort of mass spec technique where you measure the mass, you can see, oh, I got the mass of the monomer, not something that's like aggregating.
But the letting is all right.
Oh, those peptides? Well, in theory, they're like the products of proteolysis, right? So they are like along the pathway of degrading the protein. So I don't think they are necessarily supposed to be functional. They're just products of breaking down that protein. So and then your body's going to break it down further. But something about those peptides in these conditions start to just aggregate. So it's like you're like stuck in the process of just basically destroying that protein. These peptides start to inform those that protein. So yeah, so ways that people have tried to get an assessment of metal binding to these type of peptides is by trying to work with smaller forms of the peptide that minimizes aggregation. So as an example, studies have been done with a version of it that's the A beta 16. So that gives you some approximation of how these metals may be able to coordinate to the peptide itself. Again, that's like half of the peptide itself. And so here we're just going to look at some of what's understood about the metal binding. We're going to focus first on copper. So let's look at copper 2+. So we go back to our fundamentals. Copper 2+, we look up the periodic table. You get your electron configuration. That's a D9 metal. So copper 2+, typically you're talking about structure that are 4-coordinate, 5-coordinate, 6-coordinate. That's what's typical for that type of metal. So D9, if you put that into, say, a octahedral field, then we would be able to see what type of, it's paramagnetic or not. What we know is that's going to be something that's going to be paramagnetic, has a mass of 1/2. So techniques that are really useful for something that's paramagnetic would be a technique like electron paramagnetic resonance to give you some insight as to the coordination of that particular metal. So here's what's proposed. So this is a pH-dependent study done with this smaller peptide to get an understanding of how the copper 2 is coordinated. So we see that it's affiliated with 3 or 4 of the amino acids. So aspartate 1, the histidine 6, histidine 13, or maybe 14. And so coordination, hey, Oscar, what's the geometry of this copper right here? What's the geometry?
Geometry is five pyramid. I have the name. Yeah.
So what's coordinated to that copper? So we have the amino group from the aspartate. So that's like the end of the peptide. You have the carbonyl oxygen. You actually have the side group, the carboxylate from there that's coordinated to the copper at the axial position. And you have the histidines that are coordinated in the equatorial position. All right, so that's what's proposed for a pH of 6.5. Then at a pH of 9, we see a bit of a difference in terms of what's coordinating that metal. So you have aspartate still playing a role there. You have a histidine, although not clear which histidine, and then you have the alanine. So that's closer to the aspartate. And these are serving as coordination points for the copper 2, same type of coordination geometry, but not necessarily the same points of contact. Obviously, we're looking at least one different amino acid, and we also see we've lost the contact between the carbonyl oxygen. All right, so that's what observed at a pH of 9. And it's believed that for a pH of 7, we have a mixture of these two species present. All right, so copper 1. So now we have one extra electron. We're talking about D10, okay? And so now we move from something that was intermediate to something that's softer. So the typical types of structure that you will see for copper 1 would be 2-coordinate, 3-coordinate. This is diamagnetic on this metal, so that now opens up proton NMR to be able to more easily access it. I will note that with paramagnetic metals, you can potentially use NMR, but it's much more complex. Right. So these are what's proposed in terms of the pH-dependent speciation, that you have this sort of equilibrium between a 2-coordinate interaction with the copper, with the histidines. We have linear structure and there's more trigonal structure, not necessarily specified which histidines are contributing to these cells, to the two, but these are what's believed to be approximately what's coordinating that copper. All right, so this is what's proposed in the physiological pH range. That's sort of when you talk physiological, I would say anywhere down to like pH maybe, well, it depends. So if you're talking about gastric, you're really going down pH 1. But if we're talking about the cellular environment, let's say we're talking about a pH range of maybe 5 all the way up to 7.4. And that's the physiological pH range of what that would really mean. All right, so what about the other metals? So not only did we propose copper to be involved, we also proposed zinc and iron. So here is the assessment of zinc with that peptide, zinc 2+, that's D10, the type of coordinations that are possible. Zinc is what is known to be, because D10 is diamagnetic. So again, proton NMR quite helpful here. For iron 2+, that is D6, okay? So it depends. For that particular electron configuration, you can have low spin or high spin. But typically what's observed for iron 2+ are these coordination numbers. So it depends, but you can be able to use proton NMR and EPR depending on the type of the spin state of the metal. So this is what's proposed for zinc 2+. So now you have a structure that is trigonal bipyramidal. You see the points of contact to the zinc, and that's what's believed to be the predominant structure over the physiological pH range. This is what's proposed for iron 2+, and it's actually also what's believed to be true for iron 3+. Again, you have something that's trigonal bipyramidal, not exactly the same points of contact with zinc, but there are basically overlaps. It's a difference in terms of whether you have a nitrogen contact to the zinc or you have this carboxylate contact of the aspartate to the iron 2 or the iron 3. Okay. So this is just a perspective of how people have tried to gauge metal binding to the peptides to get insight as to what role do metals play as far as the ramification of this disease. But there's another layer, and that is what is referred to as the oxidative stress hypothesis. So this is added on to all these hypotheses that not only do we have the effect of the metal binding and creating maybe higher order structures, but we also have the effect of the redox activity of the copper and the iron. Obviously, zinc is not going to be in this context redox active, but the copper and the iron certainly can be. So it's proposed that these two metals may facilitate the formation of reactive oxygen species or even reactive nitrogen species. It's for the illustration of how you go from oxygen down to these reactive oxygen species. And so Dr. Scooter gave you a little bit of introduction of this by way of discussing a little bit of Fenton chemistry. All right, so this idea of these redox active metals and essentially what they can contribute to is the abundance of oxidized lipids, protein, DNA bases. So basically can lead to oxidative stress. And so I remember Javier had a question with Dr. Scooter about the ferroptosis. So this is presumably a type of ferroptosis. If you think about it, just this idea of formation of oxidized lipids via at least incorporating iron in this particular pathway. All right, so here's just the perspective of copper facilitating very similar type of chemistry as iron can do. Here looking at the formation of reactive oxygen species ROS. And so, copper can really go between copper 2 and copper 1 very easily, especially how it's maybe coordinated in the cellular environment. And so we see the formation of here, the superoxide. So how you could transition from copper 1 to copper 2 and how you generate the superoxide. So interacting with just molecular oxygen. And then how you get the formation of hydrogen peroxide. Then you have the Fenton chemistry applied here, where you get the formation of the hydroxyl radical. Then you have the Haber-Wise reaction. And so the really, really problematic one is this one here. So this seemingly simple reaction, we have RH reacting with, say, the hydroxyl radical, and then now this becomes your radical itself. So RH, if you imagine that's your biomolecule, right? That because of the formation of this radical or some of these other radicals now becomes a radical. And so you can see what can happen next more extensively. Okay. So again, this process can be facilitated by iron because of that capacity to, flux between iron 2 and iron 3. So this idea of the hydrogen atom abstraction and then leading to the radical being placed on the actual biomolecule. So you can just see what can happen further down the line, where there's the proteins and the nucleic acids. So with an excess amount of this, ultimately this can lead to cell death and then mortality if it's just something that continues to progress. Right. As it turns out, reactive oxygen species, we depend on them in our cells. So I've said this before. So it's not that reactive oxygen species in and themselves are bad. It's just that they become loss of control. And so what you have is that the rate of the molecular damage being caused by these type of species now starts to exceed the rate of the cell environment to repair. And whether or not that leads to the onset of Alzheimer's disease or whether this is actually part of the disease itself, that's still very questionable. All right, so this idea of the copper and the iron, so you form these structures, but also they maintain their redox capacity. So the question is, how is it possible for uncontrolled ROS RNS formation by plaque lead to Alzheimer's disease onset when plaque is external to the neurons and the most damaging radical, the hydroxyl radical, really cannot migrate far because it's so reactive that it will react with wherever it comes into proximity. So in vitro reactions demonstrate that the generated hydroxyl radical does not actually escape into solution, but rather cause damage to the A beta peptides itself. So that kind of shows you how it does not actually travel far. It forms and then it can actually damage the peptide from which the metals are associated. So it's believed possibly that what's occurring is that some chunks of the plaques may be breaking off, and that's probably translocating closer to the neurons, facilitating movement of these structures or the metals themselves to the actual membrane. So that's what's a possibility. So this idea that you have these little chunks that are kind of moving around, getting close enough, and then facilitating this effect on the actual cells. All right, so additional considerations. So as I was mentioning to you, Javier, this idea that those peptides are probably not necessarily, intentional as far as they just probably degradation, part of that pathway. But the thing is that something about their formation and clearance, more so the clearance, has become dysregulated, that perhaps the slow clearance is what's contributing to this onset of the plaque. Because the original cascade idea is that just the peptides themselves are aggregating. So something maybe that's regulating that process has been messed up.
So that relation.
Right. Exactly. Yeah. I mean, all our proteins and all our cells, we always have a process where proteins will reach their limit of use. We have proteases that are designed for that purpose, to break them down, and then we recycle up things, right? So you break it down to amino acids, you start using them up, and so you start generating, express more proteins and so forth. And so, yes, this process is occurring. There's something about the loss of that regulation process, and at what point is that loss what's triggering this disease? All right, so you also have, so I was focusing on the amyloid plaques, but you also have, and I haven't really paid much attention to the insoluble neurofibrillary tangles that are the consequence of the tau protein. So there's a protein that undergoes phosphorylation. That's a normal process. Many of our proteins become phosphorylated. That's typically part of signaling processing, but the issue is that they become so extensively phosphorylated that they start to aggregate. And so you get the formation of these NFTs that are occurring intracellularly. It's comparable to what's happening with the plaque. And it's possible that they may interact, these NFTs may interact with the metal ions. Right. So let's talk a little bit about some of the approach of chelation therapy to try to treat Alzheimer's disease and to think about some of the, what are those, drug design thoughts you have to take into consideration. Why this particular disease is not so easy, to control by chelation itself. All right, so stringent requirements for the design of chelated drugs. So one of the really important ones is that you need to have a chelator that does not disrupt the overall process of your body. So like, for instance, you don't want a chelator to remove a metal from transported proteins, or you don't want it to affect that iron that's getting transported throughout our body. You want it to affect that ceruloplasmin copper. So copper is a transporter, but also that's an important protein for regulating iron. So you don't lose that control either. And also you need to have something that has the capacity of crossing the blood-brain barrier, another factor to take into consideration. So here's a complication. As it turns out, so Brenda has a question. So, okay, no question. Brenda has to leave. She has an activity. All right. Well, good luck, Brenda. All right. So as it turns out, copper, iron, and zinc as assessed in both a healthy and a Alzheimer's disease brain, that the actual concentration of these metals are basically identical, right? So it's not the concentration necessarily, it's the distribution that seems to be the problem, where there seems to be a sort of a higher amount of the metals localizing in these structures. So you have a loss of the regulation of the metal as it's supposed to be more naturally distributed in the brain. So unusually high concentration in the plaques and also in the NFTs. Okay. So another important thing, the chelator must have intermediate affinity to remove the metal ions from the plaques and NFTs, okay, very important. And something we've talked about before, this idea of having like reasonable affinity, but also being labile enough to be able to release metals. But not from the important metal storage sites, such as the metallothioneins and ferritin. So you want to have a high enough affinity that you can affect the plaques, but not affect other places where metals are being stored. But another important thing that people have tried to think about is trying to restore the balance of the metal. So a way of trying to release the metals if that's even possible. So the chelator could be cleared from the brain and thus remove the metal ions, or remain in cells and maintaining the metal pool biounavailable, at least at a reasonable amount of time, so that you can start to break up the plaque aggregation. All right, so let's look at a few examples where people have tried to use chelation therapy. So this is pleoquinol. So you see the coordination sites here, label them in red. So originally, this was developed to treat human intestinal amoebiasis, which is a parasite. So it was advanced, or at least tested for Alzheimer's disease purposes. It advanced to phase 2 clinical trials, but it was actually stopped there due to major impurities in the development of the product. There's just a lot of issues with side products that were building up during the scale-up of the compound itself. So a derivative was prepared, the PBT2, that was also advanced to phase 2 trials. But here, what stopped it was that there was just not enough efficacy. What I mean by efficacy, there's just not enough of a clinical effect. All right, but let's get some perspective of metal binding for of the ligand for these metals. So here we have the perspective of coordination to copper 2+, how it is capable of coordinating to zinc 2+, and the corresponding beta values for the complexation. We have 2 of these ligands coordinated to both the metals. And so we have a reasonable affinity constants, 10 to the 10, 10 to the 8. These are pretty strong. So this was something that was measured on conditions that were trying to mimic the fluid of the brain. So a reasonable affinity constant. So some of the questions about how might this particular ligand have worked, obviously is not advancing any further, but some of the information that was obtained. So it turns out that the copper affinity is lower than that of the brain plaque. So that's an interesting thing. So the brain plaques, so those are the peptides in the plaque itself, much stronger affinity And so it seems that perhaps that this particular ligand may have served the function of an ionophore to transport free copper to neurons to try to restore the copper homeostasis. So that's what I was trying to say before, this idea of trying to restore the balance of those metals as opposed to trying to actually remove them from the brain itself. And so it may not be that the ligand itself was directly targeting the plaque, at least the copper in the plaque, but maybe was free and trying to restore the balance of that copper throughout the brain itself. Another thing is that other studies suggest that maybe this compound was not acting at all by metal binding. So maybe it was acting independent. And so it was observed that it blocked the ability of the A beta peptides to self-associate. So somehow there may have been some sort of interaction between the molecule and the peptides themselves, preventing this aggregation. And that's maybe how it was functioning, and nothing to do with metal binding itself. And that would seem likely if the first one is true, that the affinity of copper to platinum was greater than the affinity to the actual ligand itself. All right, so let's look at this other chelator, the ferrochrome. So this is an iron-binding ligand. These are the coordination sites of the actual ligand, and you see the complex. We have 3 of the molecules saturating the coordination sites of the iron itself. So this particular drug is used for the treatment of iron overload disease, particularly in this disease, beta thalassemia. So that's the complex that's proposed to form. And it turns out that complex can actually cross the blood-brain barrier. All right, so what are some of the observed properties with respect to this particular ligand, the ferrochrome? So it's actually observed to decrease the amounts of those peptides, the A beta 40, the A beta 42, and even the beta secretase, which is an important protease for cleaving A beta from the amyloid protein itself. So this was observed in a study with rabbits that had Alzheimer's disease-like condition. So something about the ligand itself was having an effect earlier on in the process and minimizing the formation, or at least maybe the longevity of the peptide, or somehow interrupting the secretase. Another thing that was observed is that the molecule had the capacity of blocking or minimizing the phosphorylation of the tau. So by minimizing that, then you would get the minimization of the formation of NFTs. So, and that was observed with the molecule again. Whether that has to do with iron binding or not, that's something that I'm not so clear on. But it could not prevent ROS generation, perhaps due to pre-binding to metals. So that's why I was saying to you that it's not clear if these functions have anything to do with being bound to iron, or if it's just the compound itself directly interacting. But it appears that it didn't actually have this capacity of decreasing the redox capacity of the iron that's bound to the plaque itself. And it may be that, let me just check. It may be a situation where that ligand, because of its high iron binding capability, is already metal bound. And so if it's already metal bound, it can have the capacity to bind the metal from the plaque to be able to have this effect of minimizing the formation of ROS generation. So therefore, unable to scavenge the metal from the plaques. So people took note of that and tried to think about, well, what to do next if that is the actual case, and maybe that we don't have the capacity of the ligand to be an iron binding ligand because it's already iron bound, right? And if you want to try to, really capitalize on that feature, well, an approach is a prochelator approach. All right, so as the name implies, is something that will have the capacity to chelate, but something needs to trigger that chelation capacity. So that's why it's a prochelator, like a prodrug, because it doesn't have the capacity in the current form. And so this here is a derivative of the ferrochrome. So that's a prochelate GL7. So let's look at the features. So you have the ferrochrome, and you have here the coordination sites of the metal. So what we observe here in this structure is you have the sugar that's basically blocking one of the coordination oxygens of the ligand itself. And then over here, you have an added feature that was built into this basically add in with this targeting group. So you're trying to do a couple of things here. You're trying to have something that blocks metal binding, but you're also adding in a feature in the small molecule to try to target the plaques themselves. Let's talk a bit about this. So you're going to trigger chelation by a biotransformation of the molecule itself. All right. So this particular derivative, this prochelate, contains that glucose protecting group. And so the idea is that now you're going to be able to transport this molecule through the blood-brain barrier without having any capacity to have iron bound to it. And then that blocking group, that glucose, can be removed by this beta glucosidase enzymes that are pretty abundant in the brain. So you're basically taking advantage of that enzyme that has a capacity to remove the sugar and now lead to the formation of the actual chelator, or at least now allow for the chelation capacity to occur. But then you have this other group here, this targeting group, that has a thioflavin T targeting group, and that basically was designed so that it has the capacity to interact with the plaque themselves. So the idea is that because of that group, it would draw the molecule towards the plaque. At least if it comes in close proximity, it would then have that contact to bring the chelator to the plaque. And there's another feature that they were trying to take advantage of was that it actually also served as a dye. So it served as a way to image, the molecule with respect to the actual product. So essentially, this was once non-glycosylated. So what I mean by non-glycosylated is once you have lost the sugar group, the GL7 can prevent metal-mediated ROS generation, and further metal ion-induced A-beta aggregation, because it now has the capacity of being able to bind the iron, but also still, because it is a metal general binder, right? It's called an iron chelator, but it still has other metal binding capability. It can also serve to possibly to bind the copper and the zinc. Okay. So any questions? Any doubts? Go ahead. Right.
Sorry. Good. So, this,
Has a higher affinity for iron. Yeah.
Wouldn't that be usually because now you will only be targeting.
I think it's trying to get both capacities. So trying to pull metal, right, from the actual structures, but then once metal bound, to try to enable the, whatever consequence of the aggregation itself. So not just affecting the metals themselves, not utilizing that metal complex potentially to affect the other. So I think let's try to restore that function as well, to use both. Yeah, because if in fact this compound has to be actually formed, and let's say that by iron, and if that needs to be formed for you to be able to see some of these other effects, I think that might be an incentive to try to restore the iron binding capacity, because it's not really clear if this is a consequence of the iron already being there. Yeah. Oh yeah, yeah. I think the idea is to try to, but that's always like a dream. It's like you try to use a small molecule that can do it all, right? But yeah, I think that's some of the idea there. Like restore the metal binding capability to break up what's already there, but also maybe take advantage of that to do some of this effect, if it's at all correlated. Yeah. Other questions? Any other questions? All right, everyone. I'll see you next time. What, you want to quiz something?